Okay, question 7. Collagen is a structural protein found in connective tissue. The diagram shows the components of a typical collagen fiber. So we have three polypeptide chains that wind together to form a tropocollagen, and then many tropocollagens form part of a collagen fiber. Explain the significance of repeating sequences of amino acids in the formation of tropocollagen. Okay, they're saying formation of tropocollagen, so therefore they're talking about this. So actually what they want is just this bit. Okay, so little will know this, but the actual amino acids involved here is mainly just glycine. If you didn't know this, then you could get away by just saying that it's an amino acids. You don't have to give them the actual amino acid name. If you did know it's glycine, then you know glycine is the smallest amino acid available, because the R group is just a H, and that would be the first mark by just saying that it is very small. And so if they're very small amino acids, then the collagen fibers will be closer together. However, if you didn't know it was glycine, you would look at formation of tropocollagen formed and you see it is very tight together. So what you would say is it will be made from small amino acids, and so once again the collagen fibers are very close together, because the smaller something is, the more compact it becomes. The more compact, the stronger, because it forms many bonds together, and that's essentially what's going on. And so you would start off by saying the glycine, or the amino acids, they are very small, and so the collagen fibers are very close together. And that will be your first mark. We've just said this. So we've said what we've seen. Now we need to explain, as it says. So what's the importance of what we've just said? Well, the closeness allows the formation of bonds. And so it will hold the polypeptide chain together. Because here, that's all bonds, because they're very close together, and so you've got attraction and you have a high chance of making bonds. And that's it. Now, which of the following bonds holds the three polypeptide chains together in the tropocollagen? Okay, bonding. Now, a process of elimination, you would get C. And, well, if you didn't know it was hydrogen, you go for a process of elimination. So ester bonds is involved in triglycerides. So that's between carboxyls and hydroxide groups. Glycosidic bonds are between carbohydrates. And peptide bonds are with amino acids. However, that is within the amino acids. So if we have this, then the other bond formed here between these two and the elimination of a water molecule, this is the peptide bond. So actually this is what's holding the polypeptide chain. So the whole polypeptide chain, it's what's holding one chain. To hold the chains together, so between here, that's the peptide bonds. However, between the other chains, you have the hydrogen bonds. And we see this in other places as well. So like in DNA, the double helix, this is by the backbone is by phosphodiester, ester bonds, but actually the helix is with hydrogen bonds as well. We see it with water molecules, and then for it to bond with another water molecule, that's a hydrogen bond. So when it's And we see it as well with proteins. So actually, like over here, so actually what holds the whole structure together is the hydrogen bonding. Right, B. Hydroxyproline and proline are components of collagen. The graph shows the effect of temperature on the helical structure of collagen with hydroxyproline and collagen without hydroxyproline. So this is without hydroxyproline, and this is collagen with hydroxyproline. We can see that this one acts better with temperature than this one. Right, analyze the data to explain the importance of hydroxyproline in the structure of collagen. Okay, so going back to what we just said is that, well, before you look at the question is, what is this graph showing us? Well, it's showing us that if we take a look at, let's see, temperature 40, at 40 degrees, collagen without hydroxyproline, we'd have 5% helical structure. But if we go 40, with collagen, we have 100, or nearly 100, like 95. So, first thing to state what we see is that the collagen with the hydroxyproline has more helix structures present than collagen without hydroxyproline at higher temperatures. So, collagen plus hydroxyproline has more percentage helix structure at higher temps than collagen without hydroxyproline. Okay, so that's what we see. So analysing it further is basically, so explain, so why does this happen? Or, make a link. So, what it tells us is that the collagen strength is dependent upon the amount of hydroxyproline present, because you normally think of collagen as strong. Well, actually, when we took out the hydroxyproline, what happened? It became weak under high temperatures. So what's this showing is that hydroxyproline is giving it thermal stability, because when we applied high temperatures with hydroxyproline, it was able to get a good result. As soon as we removed the hydroxyproline, we got a really bad result at temperatures even as low as 15. And we mentioned that. So, therefore, the hydroxyproline is responsible for giving, for holding the structure of the helix together. Moving on. The table shows information about the skin collagen of 3 animals and the body temperature of each animal. So we've got a calf, we've got a shark, and we've got a cod. And it shows us the percentage of the hydroxyproline in their skin and their body temperature. Okay. The thermal stability of collagen is described by its shrinkage temperature. This is a temperature at which 50% of the helical structure is lost. Analyze the data in the graph and the table to comment on the stability of collagen in these animals. Okay, so let's see what we have to do. We need to talk about this and the graph. And we've got something else, TS. And because it tells us it's what 50% the helical structure have lost, and the table is here, therefore we get this from the graph. So if we go back to the graph and 50%, okay? So we'll just clean this. Right, so at 50% we've got here. So for collagen without hydroxyproline, at 15 degrees Celsius, we get it's when we've got 50% structure. At collagen with hydroxyproline, we have 50 degrees Celsius. Therefore, the TS value for collagen with hydroxyproline is 50 degrees Celsius, and the TS for collagen without hydroxyproline is 15 degrees Celsius. So again, this we've just stated this over here, but what we're going to say is our conclusion again, that the hydroxyproline is giving thermal stability to the collagen, because look at the temperature difference. As soon as we put hydroxyproline, the TS value was 50 degrees. That's a big jump. And now we start analyzing the graph, because it's, the table, because it's 4 marks. 2 marks would be for the graph and 2 marks would be for the table. Right. What we can see here that calves have the highest percentage of proline, proline and hydroxyproline in the skin collagen. So therefore, their skin collagen will be the most stable. So we mention them. So calf have the most stable collagen from the other 2 options, because it's got the most hydroxyproline percentage with the collagen. So therefore we know that hydroxyproline gives strength, gives thermal stability, and so we know that it will have the most stable. And well, what's the significance of this? Well, if we now look at the other row, it says that they've got the highest body temperature. And so now we make a link. Well, if they've got the highest body temperature, they need to be sure that their collagen won't break down. And so giving more hydroxyproline would mean more collagen that is thermally stable, and so it won't degrade, it won't break down. And so the calves, they'll have a high body temperature without their skin breaking down, because they've got hydroxyproline in their collagen, so it can stabilize the helical structures. And that's the final mark, that it's necessary because they've got the highest body temperature. And that's it. The highest body temperature is the final mark, just to link everything in.